Another important thing that has to do with chromosomal inheritance is the idea of mutations. Mutations will be changes to the DNA structure or sequence which may affect the way that the organism looks like. Mutations which change the structure of the entire chromosome and change many genes all at once are called chromosomal mutations. And mutations that only change one gene are called gene mutations. And that has something to do with changing the sequence of the DNA code, not the structure of a chromosome. You see on the screen the chromosomal mutations that we learn about, including deletion where a piece goes missing, duplication when a piece gets copied twice and extends the chromosome, inversion when a piece detaches and reattaches on the opposite orientation, and you also have insertion where a piece from somewhere else gets added to the chromosome, which is what happens usually during events like translocation, where pieces of one chromosome switch places with that of another. These chromosomal mutations will change the way you actually look like, because it will be affecting many genes all at once. You also have gene mutations, which are shown on the left side. And gene mutations have to do with changing the sequence of the DNA, and those will be, for example, insertion, which adds a new base, or deletion, which deletes a base from the code and therefore shifts the entire code to the left or to the right, which is called a frame shift mutation. So insertion and deletions are frame shift mutations. And you also have substitutions, which only basically switch one letter for the another one, and that will change the base and change the protein that's based on that. And that's called a point mutation because it only affects that specific point and doesn't actually shift the reading frame of the message. Now you can also have repeat expansions when you get pieces. It's kind of like duplication, the chromosomal mutations. And these mutations may affect the way you look like. There is also a special type of chromosomal anomaly that's not actually a mutation. It's more like a problem that happens during either meiosis or mitosis, where a chromosome fails to separate successively. And so therefore, during meiosis 1, if the homolog pairs fail to separate, or if meiosis 2, the cytochromatids fail to separate, or if the same thing happens during mitosis, you end up with cells that have a normal number of chromosomes, because they come from gametes with a normal number of chromosomes, like you see in the screen. That's what happens, for example, with diseases like Down syndrome, Patau syndrome, Edwards syndrome, Klinefelter syndrome, the Turner syndrome, or trisomy X, or the XYY syndrome. These things often cause problems which were called aneuploidy, or abnormal number of chromosomes. There's two types of aneuploidy usually. You have monosomy, when you have only one copy of the chromosome type that you should have, or you have trisomy, when you have multiple copies of the chromosomes types that you should have. Examples of monosomy are Turner syndrome, when you only have one X, and trisomy will be Patau syndrome and Edwards syndrome, which are trisomy 13 and 18 respectively, or Down syndrome, which is trisomy 21, or Klinefelter syndrome, which is XXY or XXX. And all of these problems will cause major changes in the phenotype, because in the case of monosomy, it's more likely for you to have hemizygous disorders or problems because you only have one copy of the gene and nothing to protect you, kind of like it is with sex linkage. And with trisomy, you have too much DNA, which actually interferes with the way that the chemistry of the body works. And then you end up having problems like you have in Down syndrome or Patau syndrome, which causes a lot of deformities. In plants, you have an extreme of this, where every single one of the chromosomes non-disjuncts at the same time, and you get something that's called polyploidy, where every single chromosome in the set is tripled or quadrupled. And that leads to plant evolution, and I talk about that in a separate video lecture series. Klinefelter syndrome and Turner syndrome actually show that the body must do something, because when you get something like Turner syndrome that's just X, or the Klinefelter syndrome, which is XXY, and you compare that with a male, which is XY, or a female that's XX, well, there shouldn't really be a difference between them. Because in the case of the female, shouldn't the actual second X become a body anyways, or deactivate, and therefore there shouldn't be any difference between a Klinefelter person and a normal female. Likewise, if you have an XXY, the second X should become a body, which would basically make you XY or a normal male. But that's not actually what happens. And so there's an indication that the bodies must have some sort of function, because they will interfere with the phenotype in the Turner and Klinefelter syndromes. By the way, as it is suggested in this slide, the best tool to compare disorders like a Down syndrome, Patau, Edwards, Klinefelter, and all of these other non-disjunction disorders is to do a karyotype, because you will see the ester chromosome listed. And so if you actually were to do that, you will see the Patau with the 313s, or Edwards with 318s, or Klinefelter with the XXY, and so forth. and that you'll be able to compare the different disorders. Karyotypes is the best genetic tool to do that. In terms of genetics, though, mutations can be of three different types. You can have missense mutations, and these are mutations which affect the protein but do not necessarily destroy the protein altogether. It will change one little thing in the protein, usually because of point mutations, for example, which are not as bad as the frame shift mutations will be. But these missense mutations will change the phenotype because it will change the protein. And so the protein will not be the same protein that it once was. You still make a protein though. But in the case of a nonsense mutation, is that you change the protein so much, or you actually create a message that stops the protein from being produced, and you get to the point that no protein is actually made, or you make a non-functional protein. Frame shift mutations and mutations which change the code to create a stop codon, which we'll talk about in protein synthesis, actually makes this worse than normal. And those will be the worst kind of mutations there are, second to chromosome mutations, which change multiple genes at the same time. Then you also have silent mutations, or mutations which happen but don't affect the phenotype at all. How is that possible? Well, there's three different reasons. First of all, if you change the genetic code, it's possible to change the code without changing the protein. And that's because there are different ways to spell out the amino acids. We'll go back to this when we do protein synthesis. Also, your DNA has a lot of pieces which do not code for proteins. If a mutation happens in one of those useless pieces, well, they have a purpose, but they're not protein coding. If it happens in one of those node coding regions, it will not affect the protein as well. And finally, because you are diploid, even if you have one bad gene, it doesn't necessarily mean you're going to have the disorder, because the other gene on the homologous pair might protect you from actually showing the disease. And so for these different reasons, mutations can sometimes be silent. Alright? And life has these mechanisms to actually protect us from actually having problems because of mutations. In terms of cytology, mutations can happen in two different kinds of cells, in somatic cells or germ-like cells. If the mutation happens in the somatic cells, it will not affect the offspring of the person who's having it, because somatic cells are only on you. They don't actually get passed on. And so these are the mutations which will cause things like benign or malign tumors. By the way, the only difference between a benign tumor and a malign tumor is the malign tumor will actually cause problems for the person that's having it, while the benign tumor will just be kind of like a growth. But cancer is caused by somatic mutations. Now, germline mutations will change the offspring. It might cause cancer in the offspring. It might cause changes in the offspring or the evolution. So germline mutations are important for evolution because those are the ones that change the future generation. Now, if speaking of evolution, mutations can be either good or bad for you, or they can do absolutely nothing for you. So in terms of evolution, they can be deleterious if they hurt you, beneficial if they help you, or neutral if they do nothing to either help you or hurt you. For example, a new kind of coloration that makes you camouflage better can be beneficial, or deleterious if it makes you stand out. But sometimes the coloration makes absolutely no difference, and that will be a neutral mutation. Kind of like, for example, having attached earlobes or not having attached earlobes in humans. But what kind of things cause mutations? Well, you can be exposed to radiation or to chemicals which cause mutations, which are called mutagens, especially those that cause cancer, which are called carcinogens, like the ones that we find in cigarettes. Or mutations could also cause by systematic problems in protein synthesis or DNA copy, or during the cell division process. You can also get mutations because of invasions of pathogens, which try to attack your body, go inside your cells, and cause changes to the cells. But why is it that some people get more mutations than others? Well, first of all, your immune system is supposed to protect you from actually getting these mutations. Whenever a problem happens in your cell, some cells actually have mechanisms to auto-destruct. It's called apoptosis, if they notice that the cells are actually defective. And if the cell is defective, body cells will actually attack it and go over the cells and consume them to destroy them to prevent them from happening. But certain things can actually deplete the immune system and increase the exposure to causes of mutations and make you more likely to have problems leading to mutations. And that involves, for example, being young or old when you have a compromised immune system, having a poor diet, having exposure to the causes or lack of exercise, not enough water, sunlight, or air that's of good quality, too much stress, lack of rest, having excesses on your life. Too much of anything is a bad thing. Or having any kind of immune disease like HIV or other autoimmune diseases like lupus. On the other hand, to actually make you less likely to get a mutation, you should do, for example, the New Start. Have good nutrition, good exercise, good amount of water and sunlight. Have temperance instead of excess. good air, rest a lot, and trust something greater than yourself, even if it's to trust in yourself and the power of yourself or the power of a higher being. These things have been proven to increase your life's chances of having less mutations and having problems such as cancer. Cancers, by the way, are caused by several things, including problems with cell division control, problems with cell communication leading to the formation of defective proteins, and the proliferation of chemicals which induce the cells to go into that state. But a tumor is not necessarily going to be cancer. The difference between a benign and malign tumor lies in whether or not you are displacing or affecting an organ or producing toxins which are destroying the body. Now, malign tumors have a tendency to actually spread in something that's called metastasis, which is a serious problem. But we get thousands of these mutated cells every day. The reasons why we don't get cancer as often is because these cells are attacked and self-destroy, or they get attacked by our own immune system. But if you have problems with the immune system or great exposure to causes of mutations, it's going to increase your chances of actually having cancer. And some people actually have genetic problems which lead them to have cancer, or genetic reasons why these cells go into this hyperdrive mode. It's so hard to cure cancer though, because the genes that cause cancer are pleiotropic. They actually do other jobs in the cell, so you can't just delete them. And the cells, every time you find a cure for one kind of cancer, a new kind of mutation happens and a new kind of cancer shows up. And so we're never going to get rid of cancer. Cancer is part of the process that actually creates life. It's part of the changes that create mutations in evolution. And so the same mutations that sometimes makes us evolve can also cause cancer. And so you can never get rid of cancer. So since it's so hard to cure, the best way to handling it is trying to avoid the things that cause mutations and to actually catch it early, to prevent cancer with a good new start kind of life, and by doing routine scans which kind of try to detect that cancer. And then, as long as you catch it early, you can start treatment early. And modern advances in biotechnology, which we'll discuss when we do that lecture series, which include personalizing treatment with DNA-based research and also delivering the medicine directly to the cells that have cancer with new nanotechnology that will make it easier for people to actually survive cancer. The last thing we have to talk about in terms of inheritance and chromosomes is the idea of blood types. Now, of course, blood types are just one of the representations of human phenotypes, and they're caused by a multiple allele thing. There's an allele for A type, which is basically the surface protein presence in the red blood cell in the ABO system. There's actually many different types of surface proteins in red blood cells, and you see some examples of them shown here. The ABO system is the most famous one, along with the RH system, which is having or not having the D type of surface protein of the rhesus factor, which was discovered in monkeys. Now, scientists were studying the transfusions and trying to figure out why sometimes you had problems with transfusions and why other types you don't. And they figured out there were different kinds of surface proteins in the red blood cells. The original type, according to simian studies, is the A type. But a missense mutation happened that changed that into the B type at some point, probably somewhere in Asia. A B type will be then a combination of A and B type when that happens genetically, and that's a co-dominance thing. But A and B are dominant to the O type that evolved last among the people who were then waiting on to become the Native Americans. And that's a nonsense mutation that basically made the cell not have surface proteins. Anytime you don't have surface proteins, you're going to have antibodies against the people who are different from you, which means the A type has antibodies against B, and the B type has antibodies against A. Since AB has both antigens, you cannot have antibodies, and since the O type has no antigens, it's going to have antibodies against both A and B. That helps us understand why the donation patterns work. A basically can donate to AB or to itself, since AB can then receive from everybody since it doesn't have antibodies. O, on the other hand, can donate to everybody since it doesn't have antigens. It doesn't have surface proteins to generate a reaction. Which means everybody can receive from O and everybody can donate to AB. But O can only receive from itself. And A can only donate to itself or AB, and B can only donate to itself or AB. And a similar thing happens with the Rh factor of plus or minus. And that's blood types. So chromosomal inheritance has advanced our understanding of genetics, and there's a lot yet to do. Our understanding of genetics has only just begun. We continue on our journey to try to understand where the genes are, what they do, and how they are passed on. So get out there and don't do anything that wouldn't make your mama proud.